Good afternoon, my name is Stephanie Uervides and I will be presenting on Chapter 44, Genetic Controlled Vectors. So before starting this chapter, I wanted to give a little bit of a summary of the topics that will be discussed. For example, population suppression, sterile insect technique or SIT, the challenges associated to these techniques, the release of insects carrying dominant lethal alleles or RIDL, hybrid sterility, and population replacement. I also wanted to give a brief history on the beginning of these studies, which started in 1944 through 1945 with F.L. Vanderplank, who was able to eradicate the sleeping sickness in Tanzania through the use of interspecific hybrid sterility, which is when two species interbreed and produce sterile hybrid offspring. Vanderplank employed the notion of interspecific hybrid sterility in the attempt to combat sleeping sickness that was experienced in Tanzania, which was carried through the vector Glossina swinertoni. So basically what he did was he employed the non-vector species known as Glossina morsitans centralis, both of these being tsetse flies, and ultimately the mating between these two species led to the birth of sterile hybrid offspring. So the male hybrids were noted to be fully sterile, while the female hybrids were noted to be partially sterile. Ultimately, Vanderplank took the first steps towards defining the population suppression notion that we will be discussing throughout this chapter. Population suppression is the employment of strategies aimed at reducing the density of a specific population of vectors. It aims to decrease the number of disease-transmitting organisms, and it's an active area of research. Methodologies in which population suppression occur is through something known as sterile insect technique or SIT. Sterile insect technique or SIT is a topic that is heavily discussed throughout this chapter, as based on operational effectiveness at an extensive practical scale, this is the only genetic approach with major success. Basically, it consists of releasing target pests that have already been sterilized back into their natural habitat. So in the image to the right, we can see how these mosquitoes undergo mass rearing. Then after being reared in the laboratory, they undergo sex separation and undergo irradiation, which causes the sterility to occur. They are then released back into their natural habitat where they are expected to mate with the wild female and cause them to give birth to sterile offspring or no progeny at all. SIT methodology consists of releasing insects that carry dominant lethal mutations in their reproductive cells. They obtain these dominant lethal mutations through treatment with ionizing radiation or chemical mutagens. Any wild female mating with the released males do not reproduce and only lay sterile eggs. The effectiveness of these SIT techniques stems from the species themselves. In order for these species to be good candidates in receiving this technique, species should be easy to rear in laboratories, cost-effective to maintain in these laboratories, and dominant lethal mutations need to be able to be readily induced. The image to the right of this slide represents a figure that was included within the chapter, basically summarizing the image that I previously included in a few slides ago, where mass rearing of a target species is induced at an insect factory. Then they are separated through their gender. The males are specifically reared and undergo sterilization through chemical mutagenesis. They are then released, then they mate with the wild insects, and they reduce the reproductive potential, ultimately leading to either population suppression or eradication. SIT success heavily depends on the sterile male's ability to locate, attract, and mate with wild females, as there is an innate competition with the wild males. Competition between sterile males and wild males makes it imperative for sterile males to be released in quantities that exceed natural population densities by at least 10 times. Therefore, SIT releases are usually preceded by insecticide treatments, and this is done to minimize the initial populations. Although the main limitation is the competition that is seen between the two males within the community after the release of the sterile males, other factors that influence SIT success are residual fertility, relative competitiveness, immigration of fertilized females, reproductive capacity of the species. Two species that have been very successfully treated with SITs are the screwworm fly and the tsetse fly. From 1988 to 1991 in Libya, North Africa, the screwworm fly were noted to be seen in animal wounds. They were previously limited to the Americas, so they were known as the New World screwworm fly. However, they were probably introduced through contaminated livestock to North Africa. These unusual maggots were seen to affect multiple hosts such as camels, cattle, horses, sheep, goats, donkeys, and even humans. Every 3 weeks, there was a new generation that was expected, and uncontrolled movement of livestock were expected to spread the disease even more widely. So, due to economic and social threat that was imposed through these maggots, the Libyan government and relevant UN agencies began a plan against the maggots. The plan consisted of 4 different steps. The first, surveillance to identify the limits of distribution. Second, treatment of wounds on infected animals and humans. Third, the spraying of livestock with insecticide. And fourth, the release of large amounts of sterile flies. SITs were employed because that is the methodology that was used in America, where without it, the screwworm flies would not have been able to be contained. The screwworm fly was bred extensively, and about 500 million sterile flies per week were produced. The releases began in December 1990, and by May 1991, no new screwworm fly infestation cases had been confirmed. Despite this, however, the SIT releases continued up until 1992 to ensure complete elimination. So overall, SIT is the most potent control method when applied intensively over a wide area. Without SIT, there would have been no screwworm eradication. However, it would not have worked on its own and must have been applied in combination with other efforts. Therefore, it needed to be part of an integrated program. Ultimately, despite SIT being expensive, its effectiveness counterbalances the cost. Another prime target of SITs are the tsetse flies, which are a vector for trypanosomiasis. They are an ideal target as they have low natural fecundity and they have poor potential for population recovery. To treat the trypanosomiasis seen in Zanzibar, East Africa in 1994, SIT efforts were employed. The methodology that was associated with the SIT methodology consisted of aerial releases of 8.5 million tsetse fly individuals that began in 1994. Alongside these individual releases was an insecticide treatment. In 1997, species had been eradicated from the island. When the book was written, success was seen in Zanzibar, and this was used to organize a campaign against tsetse flies in Ethiopia, and was currently being employed to eradicate the flies from a 2.5 million hectare area. So as we have been discussing, SIT consists of the dispersal of large amount of insects that have been exposed to chemicals that have led them to be hybridized and sterile into their natural environments. Therefore, since the release of large amounts of these insects are conducted through SITs, there were a lot of challenges that were experienced through the use of this technique to control sleeping sickness. This is because sleeping sickness is transmitted through tsetse flies. The release of tsetse flies sterile or not, is impractical because regardless of their gender, they are still biting human hosts, as male tsetse flies also feed on blood. Therefore, this increases the risk of disease spread, making it impossible to reduce biting by only releasing males. An alternative to this process of SITs is auto-sterilization. This would be a theoretical approach where the chapter basically discussed that experimental traps could be designed to attract the flies and chemically sterilize them, and they would then be released back into the wild. So SITs have been noted to be more effective when only the female of a vector species takes blood. Therefore, mosquitoes are a potential target of SIT employment. In 1970, a field trial was conducted in Seahorse Key, Florida against Culex pipiens quinquefasciatus. The methodology of this field trials consisted of releasing on a daily basis 8,000 to 18,000 sterile males. The results consisted of having the species eradicated within 12 weeks, or 6 generations of the species. However, this field trial was limited as these results were only temporary due to reinvasion that occurred from fishing boats and other external sources. Additionally, there were a lot of public concerns that were associated with this field trial due to the use of chemicals. The use of chemicals to sterilize mosquitoes caused concern of contamination from the released mosquitoes. Therefore, researchers sought other methods, such as the chilling of mosquitoes and the releasing of these mosquitoes. They are released at a 10-to-1 ratio. The outcome of this second part of the study consisted of complete elimination once again, but reinfestation continued being a challenge. To build upon the notion of having to deal with an issue such as reinfestation, we will now be discussing other more important technical challenges that are associated with using SITs in mosquitoes, such as automated mass rearing. The issue with this is having to develop efficient and large-scale systems in order to breed in large amounts in a cost-effective manner. Second, genetic sexing. An issue with this is having to separate between genders before release, and this is in order to not increment the increase of biting/disease transmission. Third, rapid sterilization, which would be the employment of and discovery of fast sterilization methods. Fourth, quality control, which would be having to ensure sterile mosquitoes are fit to compete with wild populations. Fifth, transportation release, just having to deal with the employment of safe transport and effective distribution of sterile mosquitoes in target areas. And sixth, trapping to evaluate progress in order to establish monitoring systems to measure the effectiveness of the programs overall. So out of the 6 challenges that I previously discussed, we will be placing specific focus on 2 of them. The first being automated mass rearing. Currently, mosquito rearing does not exceed 7 million per week. Meanwhile, screwworms are at 500 million per week. Natural mosquito densities are much higher than those of screwworm flies. This is because there are a lot of key challenges that are associated with factory breeding of mosquitoes. For example, they require efficient mating, feeding, resting, egg laying, larval development, and pupae harvesting. Meanwhile, the adult mosquitoes also need to be monitored heavily, and their behaviors must be carefully contained, such as feeding, watering, egg collection, in order to prevent escapes. So overall, mass rearing in factory settings is extremely convoluted and must take into account a variety of different requirements each vector may have. The second challenge we will be discussing more in depth is genetic sexing techniques. Safe SIT use consists of only releasing male vectors in order to prevent vector-borne disease spread in humans. So either there needs to be an avoidance of production of females, or females need to be eliminated as soon as possible within the factory production line. Therefore, GSTs or genetic sexing techniques for each species are needed. Most successful SIT trials enacted GSTs. This was seen in Anopheles albimanus and Drosophila melanogaster. A successful SIT trial was seen in Anopheles albimanus, which underwent translocation-based techniques. Sexing in this species was achieved through the exposure of the eggs to an extenticide. The insecticide depended on the translocation of the resistant gene propoxer onto the Y chromosome. Therefore, it depended on the presence of the Y chromosome seen only in males, making only male eggs survive the exposure, and the outcome consisted of the population being 99.9% males. Meanwhile, Drosophila melanogaster underwent recombinant DNA technology, also known as the release of insects carrying a dominant lethal allele or RIDL. Sexing was conducted by involving a dominant lethal gene that is typically suppressed by tetracycline in the diet. When the gene is active, only female larvae are killed. Therefore, tetracycline-free diets were employed to prevent female offspring from being born. RIDL has a lot of potential as it has been seen to be successful in species such as Drosophila. Therefore, it can be applied to other insects with medical importance. As this is the introduction of transgenic stock into wild population, public opposition is expected. Despite this, however, it still is a very potential source of research as it is a more cost-effective method since the insertion of lethal genes at multiple times in the genome would eventually produce male-only offspring. And multiple insertions of these lethal alleles have a more confounding result. For example, a single insertion leads to a ratio of 1 female to every 2 males. Meanwhile, 6 insertion leads to 1 female to every 64 males. Ultimately, the effect of population decline would be greater than SIT at the same release rate. Hybrid sterility is the process in which offspring that cross between closely related species or subspecies become sterile. This phenomenon occurs in several medically important insect groups such as mosquitoes and tsetse flies. However, the effectiveness is species dependent. An example of a species that has been exposed to hybrid sterility research is the Anopheles gambiae complex, which is the vector for malaria. Hybrid sterility was hoped to be successful in controlling the population of these vectors. However, favorable results were not yielded. In 2025, there is still an active area of research. Over the past decade, special focus has been placed on genetic, cellular, and biochemical processes associated with hybrid sterility. In 2019, Liang et al. Published a paper investigating the cellular causes associated with male hybrid sterility within the complex, and in their paper they stated that the hybrid sterility is associated with germline stem cell arrest, meiotic failures, and genetic incompatibility seen. Another species that was exposed to hybrid sterility research was the tsetse flies, which were the vectors for sleeping sickness. Within this species, hybrid sterility was effective, ultimately revealing that hybrid sterility is a promising area of research. An alternative to population suppression is population replacement, which is the prospect of replacing disease vectors with harmless forms of the same species. It is the genetic structure of pest populations being modified in a way that favor human benefits, and this is done through the use of useful genes and transporting mechanisms. Useful genes are genes that prevent disease transmission, and they do so by altering vector behavior or producing physiological changes in parasite/vector interactions. The goal of WHO is to transform malarial vectors into harmless insects that do not transmit diseases. In 2018, a study was published utilizing CRISPR-Cas9 gene editing system, and they used this to target the double-sex gene in Anopheles gambiae. The female's transcript is highly conserved, and it has a highly conserved exon called exon 5. The boundary found between intron 4 and exon 5 was disrupted utilizing CRISPR. Neither male development nor fertility was effective, but homozygous females became sterile through this gene editing. Highly conserved nature of the exon 5 boundary caused a 100% prevalence within 7 to 11 generations of this species. Ultimately, this led to egg production to be decreased and the population to collapse. The proper use of useful genes can only be done through the use of transporting mechanisms as well. which refers to a genomic element that is linked to the useful gene that allows it to spread throughout the wild population despite selective pressures. Several possibilities have been considered of becoming reliable transport mechanisms, such as negative heterosis, meiotic drive, and transposable elements. In negative heterosis, cross strains are less fertile than when bred singly or when hybrids are sterile/partially sterile. One strain would eliminate the other, ultimately becoming a potential population replacement method. The theoretical advantage that this has is that it offers a way to exploit the natural fertility reduction between specific strains of mosquitoes, potentially removing the need for costly artificial sterilization techniques. Meiotic drive is a phenomenon where one allele of a pair is passed on disproportionately. Meiotic drive can be used to increase the frequency of certain genes that are targeted. By using this meiotic drive, scientists could increase the spread of traits that prevent disease transmission more quickly, or if linked to a beneficial gene, meiotic drive could help spread traits that make mosquitoes resistant to carrying diseases like malaria, and this could replace the need of regular releases of sterile males or other control methods. Transposable elements can move genetic material between populations, which can be used to introduce beneficial traits into wild mosquito populations, and it's a powerful tool in genetic modification, especially if linked to a disease-preventing gene. Researchers could potentially insert genes that are not only carried within a population but could spread between species, enabling a more robust and widespread solution to vector control. And these are my references. Thank you.